So hello everyone. Today we're happy to welcome Dr. Marion Eckmiller that is going to talk about some theoretical studies she has been doing of the retina and especially like how the cone signals are working for lens accommodation. We're very happy actually because usually in the talk series we're talking about neural computation in software and hardware, and I'm also like very keen to hear about neural computation in the wetware. So just a brief introduction of Dr. Marion Eckmiller. She has been carrying undergraduate studies at UC Santa Barbara in biology, did a brief sojourn in Scotland during the undergrad studies. Pursued like continue her studies in graduate studies at Lafayette, West Lafayette University, Purdue University, sorry, in West Lafayette. Then end up having a PhD in biological science at UC Santa Barbara, and carried a postdoctoral work at UC San Francisco and the medical school with a fellowship from the National Eye Foundation. And after that, like she moved to Germany and spent the rest of her career at the CNO Vogt Brain Research Institute in Düsseldorf, where she ended up leading a group on retinal cell biology. And based on all her work, now she has been retiring and she's living in San Diego, but is still associated with the Düsseldorf University, where she spent most of her career, and has been doing these theoretical studies on the signaling of cones, sorry, mostly in San Diego, like based on previous research over the years. So without further ado, I will let Dr. Eckmiller like explain all of her theoretical studies that she has been carrying in. Thank you.
Thank you for that kind introduction. I'm very grateful to be able to give a talk in this series at UCSD for the invitation. I am really excited to present my findings, recent findings about evidence for a novel specialized area within human macula in which cone signals specify ocular focus for lens accommodation, the focal annulus. So we are all familiar with the human fundus. The optic nerve head is a blind spot because there are no cones. The macula in the center of the fundus has macular pigment. The smaller fovea is highly specialized within the macula. And on the upper right, we see the visual acuity of the human eye as a function of eccentricity. The acuity is very high in the center of the fovea, decreases exponentially. This is related to the density of cone photoreceptors, shown in the lower right, which peaks in the center of the fovea and decreases exponentially. The fovea extends plus and minus 2.5 degrees eccentricity and is bounded by these two lines, vertical lines. So on the lower left, we see the beautiful work from Christine Curcio in showing the density of cones in the color code, with the fovea being this white dotted area. And really in the center of the fovea, there is the foveola, and there the number of cones, the density of cones is particularly high. The work about cone directional alignment was started by the accidental discovery by Stiles and Crawford. They described in 1933 that light entering the human eye, a beam of light appears very bright if it comes through the center of the pupil, but if it enters the eye, the same light from an eccentric part of the pupil, the light appears less bright. The magnitude of that effect is shown by this graph, a Gaussian function. Here is the x-axis as the point of entry of the light in the eye. So it seems that the best light effect occurs in the center of the fovea. And this has now come to be known as the styled Crawford effect. And it is believed to be caused by the fact that individual cones point their long axis towards the center of the pupil, as shown in the upper right. Now, normal cones in human eyes do not point towards the center of the globe. So I have been interested in this mechanism of cone directional sensitivity and alignment. And as you will see in the left, all of the cones are believed to be pointing towards the center of the pupil. And an individual cone cell has many components. The cell nucleus is here within the dense retinal tissue, which ends at the external limiting membrane. And after, peripheral to that, there's an inner segment and an outer segment, both of which are in the subretinal space, and thereby the cone can change its direction of alignment by bending here near the ELM at its optical aperture called the base of the myoid. So the cone needs to remain straight and rigid for this part that bends. Let me say the cone connects to the next cell, the bipolar cells, via its axon, which is coming out here. We're going to talk about this inner axon quite a bit. So in 2004, I proposed that the alignment of an individual cone to light is controlled by a sensory motor negative feedback mechanism mediated by its microtubule-based intracellular cytoskeleton. The direction of a cone is adjusted to match that of incident light so as to maximize its light catch. In effect, such a mechanism renders a cone sensitive to the vergence of incident light, so to the direction it's coming from. So in 2004, I published a paper called proposing that defective cone cell cytoskeleton and cone alignment can lead to energy depletion and cone cell death in macular degeneration. This was after it became known that macular degeneration can be caused by defects in genes related to the cytoskeleton of photoreceptor cells. So we don't have time to go into the details, but I believe that if the cones are not pointing correctly because something is disabled with their mechanism, they're going to keep trying and they're going to run out of metabolic energy because they're not accomplishing their task. However, you could ask if all of the cones in the retina have the same cytoskeleton, the same genes, why is it that only the central macular cones die in macular degeneration? So I became more interested in the specific specialties of the central vision of humans. And in 2008, David Williams, while in UCSD, working with Don McLeod, published a paper which was very important called The Visual Consequences of the Foveal Pit. He measured the Stiles-Crawford effect in the human fovea and found, as shown on the left, that as a function of eccentricity, the cones, if they all pointed like they're supposed to, based on the Stiles-Crawford effect to the center of the pupil, their magnitude of the Stiles-Crawford effect would be this oblique line. However, he found that that was not happening in the fovea, but in the fovea, cones at this eccentricity were pointing to a different direction, and cones on the other side of the fovea at the same distance from it were likewise pointing away from the center of the pupil. And this was very unexpected. These findings appeared to contradict the Stiles-Crawford effect. However, he suggested that these foveal cones do point correctly towards where they're getting the light, but that their light has been shifted in the pupil because it has the light coming into the eye has been shifted by light refraction at the sloping walls of the foveal pit. So I was intrigued by that finding and have been trying to understand it. Since that publication, other research groups have also found reduced Stiles-Crawford effect in the central retina. So let's look at the fovea. In fact, in 1942, Walls proposed that light coming into the eye, when it hits the sloping walls of the foveal pit, will be refracted in the retina, which is denser, and that this will result in a magnification, magnification of the size of the image on the retina. And okay, that's similar perhaps to what Williams had found. So we can ask, is the fovea important? People have said the fovea is the most essential part of the retina for human vision. They have also said it is the most valuable 1 square millimeter of real estate in the human body. However, in 2008, Mike Marmer from Stanford published a paper called Visual Insignificance of the Foveal Pit. So we can conclude the human fovea is not well understood. At the bottom, we see a drawing of the cell layers in the fovea of humans. It comprises several layers as usual for the retina, but many of the layers are displaced laterally by the formation of the foveal pit. So there are ganglion cells and amacrine horizontal cells and bipolar cells, and the cones and rods are down here at the outer part of the eye, and we see the individual cone outer segments. We see the external limiting membrane, and the cone cell bodies connect via their inner axon to bipolar cells in the Henle, forming the Henle fiber layer. So there is only at the fovea this thick layer of oblique axons. Okay, so what are the special features of the central high acuity vision in the human? There are macula and the foveal pit, making the eye, the retina particularly anisotropic, so that we must make frequent eye movements to project visual objects onto the fovea. So what are the sequence of steps? You would have heard, we first foveate a visual object by moving our eyes. We bring it onto the fovea, and we then fixate the object and really pay attention to it. And then if the object or our head moves, we can follow it, the object, making saccades and smooth pursuits. Now, however, that really leaves out a really important step, which is focusing on the visual object. Why have people not thought about that? Well, in large part because the sensory signals that are used to control and inform lens accommodation have not yet been identified, remarkably. So ciliary muscles are used. That's known. Most of the circuit is quite well understood. But the first step, what cones are involved and how they do it, has not been identified. So I was trying to understand how cones align. And what I sort of have stumbled upon, I believe, is how the eye focuses. One knows that cones in the fovea are probably involved in this. And I have found that there is a subset of the cones in the fovea that are able to do this. Okay, so I was following up on Williams' study in 1980, and I hypothesized that the specialized structure of the macula confers optical properties that influence cone alignment, enabling some cones to specify ocular focus. So we're talking about the foveal pit, the Henle fiber layer, which contains bundles of cone inner axons and is birefringent. By OCT, the human macula, the HFL layer is not distinguishable. So here is a drawing of the flattened macula. And again, the Henle fiber layer is shown down here. So the steps in a light tracing analysis that I made were 1, to construct a macular template, 2, to calculate light refraction at the inner limiting membrane, and 3, to calculate light double refraction as it enters the HFL, and then the light leaving the HFL is going to bend backwards and travel through the optic nerve layer until it reaches the optical apertures of cones. Please realize that in the fovea, all of the light that reaches the optical apertures of cones must travel through this Henle fiber layer. Okay, so here is a section, LM histological section through the center of the human macula, kindly provided by Anita Hendrickson and Christine Curcio. And I have studied this and other sections. And here is a diagram of the relevant structures. And you will see as a function of eccentricity, the location of cone inner axons. Those are what we're really interested in here in the Henle fiber layer. And we have analyzed the orientation of those axons and the thickness of the layer. And there are also other cone outer axons, but there are not very many of them, and they're really in the center. So this drawing was used to show, to measure the orientation of the cone inner axons. And I took that as the crystal axis for the birefringent Henle fiber layer. So here is the macular template that I constructed. It is highly simplified, two-dimensional template. I've assumed that retina fovea is radially symmetrical. We've flattened it. And all these dimensions were taken from LM measurements and from published these days in vivo OCT images of the human macula. Okay, so I took measurements. So this work I actually was starting in Germany at the Vogt Institute. So I took measurements at the 13 eccentricities shown here. And we took the vertical and horizontal axes are shown at the same scale, which simplifies calculation of angles. I use sigma plot. Okay, so light comes in, so the light comes vertically in because we flattened the retina, and we're going to trace its path going through the inner retinal layers into the Henle fiber layer, through the HFL, through the ONL, and then finally it can reach at the ELM, the optical apertures of cone photoreceptors. So what did we find? First of all, light refraction at the ILM can be calculated using Snell's law. That is relatively straightforward. And I am not going to give you all the details, but what is important is to calculate the change in the light direction, okay? Refraction is always calculated with respect to the normal angle to the surface, which I had to get used to. Okay, so we have to calculate how the angle here that the light is making coming in with the ILM surface, how much it's going to get bent, which would be this amount by theta 2. And then the important thing is the difference. How much has the direction of the light coming in been shifted by refraction? So the important parameter is the change in light direction that results from that refraction. On the right, I show you the results that we found. And as a function of eccentricity in the fovea, the change in light direction. Okay. And what we found was that at high eccentricities and in the central of the macula, there's no shift, but there is a maximal shift at 1 degree eccentricity. So it's a little bit gradual. And the maximal shift is only of 0.32 degrees of visual angle. This is quite large, but it may be visually significant. Okay, so now we're getting down to business. How do we calculate double refraction? So double refraction is this strange phenomenon that occurs in certain natural crystals and man-made materials that have highly ordered structures. They are optically anisotropic, and they can be birefringent. So here is a calcium carbonate or calcite crystal that exhibits double refraction. If light goes in through it, you will get two images. Double refraction produces double images. So in the center, here is a birefringent crystal. Unpolarized light, when it enters this birefringent crystal, will get split into two separate orthogonally polarized rays of light. So one ray of light is going to be called the ordinary ray. It obeys the Snell's laws of refraction, and it does not, in fact, change its direction if it is traveling along the orientation of the crystal axis of this crystal, the optical axis. The other ray, which this is half and half, it's a 50-50 deal. Half of the amplitude of that light becomes an extraordinary ray, an E ray, which does not obey Snell's law. So it will get refracted. And you also please notice that the light that exits in these two displaced rays is polarized. And the two kinds of rays are polarized orthogonally to each other. So we know that the HFL is a positive uniaxial birefringent crystal. And that made me ask, how on earth can the fovea provide high visual acuity if light passing through the HFL produces double images? Seems very strange. So how do we calculate this? So double refraction is analyzed usually using what's called a refractive index ellipsoid or indicatrix. So on the right here, you see this ellipse. That's going to be defining or helping us to know, calculate the path of the E rays. For simplicity, let's first look over here on the left. The O rays in blue, they follow Snell's law. So if this is the interface going into a birefringent crystal, the incident light comes in here, and depending on what the crystal axis is of the crystal, it may be, I mean, if that incident light happens to be coming in along the crystal axis, then that ray will become an O ray, and it will continue going in this direction. But depending on the orientation of the crystal axis, the O ray will be coming in at different directions. So it may be an E ray. So the E ray, so the direction that the light goes depends on the refractive index of the medium. So the refractive index in a birefringent crystal depends on how the direction of its crystal axis compares to the direction the light is traveling at. So it can, the refractive index within the crystal varies from the N for the O rays, which in this case is a small number, to the maximal refractive index for the E rays, which will be down in this direction if if the crystal axis is in this direction. Now, in the HFL, the surface here of the interface going into the HFL changes at every retinal eccentricity. So this ellipse, its axis is going to have to be changing all the time. And the crystal axis within the HFL also changes at every eccentricity. And we have to figure out how to do this. So the the refractive index is going to be defined or by where we come in onto this ellipse. So it can be just the n sub O, or it can be n sub E, or its maximal n refractive index would be down here. So how can we calculate the path of light through a birefringent HFL? This requires knowing the relevant geometry and the values of both n sub 0 and n sub E. All right, what do we do now? Fortunately, Barry Sensi in 2013 studied the retardation of light passing through the human HFL using polarization sensitive OCT. And he found that often the most retardation, which is related to birefringence, occurs as a ring around the fovea, shown here in color coding. And he measured this in various humans' eyes, the retardation as a function of retinal eccentricity, and his data are shown as these red dots. And I compared that retardation as a function of eccentricity to the thickness of the HFL, which are these black dots. And I found that fortunately, these two values could be scaled such that they pretty much overlapped each other. One can use that information to say that 50 microns worth of HFL thickness gives you about 19 degrees of double phase retardation of the retardation of the light. Okay, so when you do the calculations correctly, you come up, if you know with what you're doing, with a great finding that the index of refraction of the retinal tissue, which was normally 1.36, gets increased by a great large amount of 0.00044 to arrive at the greatest index of refraction for the E-rays is going to be something like 1.3604 units. Okay, so it's not very big, but it's there, and I think if it's there, it's doing something. So I was able to consult with Martha Rosetta Aguilar in Mexico, who was very helpful and explained to me how you can calculate by double refraction, not using the complicated Maxwell's equation, but using the what I find simpler Huygens principle, which is based on wave part of light. And you can then define using generalized condition, all of these factors, which you have to calculate in a consistent manner. So everything changes with respect to everything else. And you've got to keep track of that. And if you're doing it correctly, you can then use the equations shown here below. The refraction angle for the O ray winds up being just the direction that it came in at. But the reaction angle for the E ray is this nice little formula here. So within the HFL, the E ray is going to be refracted very much because the HFL is what is birefringent. So this whole calculation would give us what angle the light is going within the HFL, but that is not a fair criterion for light going through the whole fovea macula. We have to multiply that theta prime of the E, which is here within the HFL, by the relative thickness of the HFL. So if we multiply that by the thickness of the HFL divided by the thickness of the retina, we get the comparable fair sort of calculation of what the direction that E-ray is going to be going through, how much it's going to shift. So again, skipping many steps, on the left, these here is shown the final results for the E-ray, the change in light directions of the E-ray, shown here, the change in direction as a function of eccentricity. If we look just within the HFL, the maximum shift winds up being conveniently occurring at 1 degree eccentricity, and that is a shift of something like 0.55 degrees. And the maximal shift of the E-ray within, I mean, from across the entire ILM to ELM, it is only about 0.34 degrees. So we have to reduce it down to here. So when we compare that and the E-rays and the O-rays and put them together on the same graph, we see on the right that the O-rays and the E-ray change in light direction are hardly different from one another, but they're both maximal at a 1 degree eccentricity. And that on the whole retina, fovea, forms an area that is shaped like a ring or an annulus. So this is located where the slope of the fovea is maximal. So one important thing is that the O-rays and the E-rays were orthogonal to each other. What does this mean? So in 1619, Christopher Scheiner, a Jesuit priest in Germany, was the first person to demonstrate the focusing ability of the human eye using a simple device called a Scheiner disc. This was an opaque disc with 2 holes in it, and he put that in front of people's eyes, and he had shown light into their eyes. He actually used white light, but if the disc is at the right distance from the eye and the light is coming from infinity, the end of the eye is emmetropic and in focus for that light, then the light will form one spot on the back of the eye, namely on the fovea. If, however, the eye is myopic, then 2 spots will be formed on the fovea. And if the eye is hyperopic, 2 spots will be formed, but the light will not have been crossing itself, or the light coming through each of these little apertures. So you can figure out from this whether an eye is myopic or hyperopic, and at what focal level you can make it emmetropic by putting a lens in front of the eye. So I was then realizing, just learning about this, that a Scheiner disc with 2 apertures could be made into a Scheiner disc with 6 apertures. And I said, oh, wow, that is just like what the fovea is doing. We have in this annulus, it represents a Scheiner disc with continuous or multiple apertures. So I concluded that refraction of the eye can be determined using a Scheiner disc or this annulus in the fovea. What does that mean? Well, it can mean that cones in this annulus of the human fovea can detect, well, we know that they can detect light vergence by their alignment, and that they then will align, if they're in the annulus, to the light that has been shifted by refraction and double refraction at the fovea pit. And they can then use these cones, the Scheiner principle, to specify and to monitor ocular focus for lens accommodation and for emmetropization, balanced growth of the human eye during development. So let's give a name for this special annular region. So I coined a new term, oculomonitor, for this, the function of this region. So it's going to monitor the eye focus, in fact. And I was then able to call this annulus the Foveal oculomonitor cone alignment locus, which conveniently resulted in focal annulus. Okay, so to recap, the previous model was that understanding all cones are aligned towards the center of the pupil of the eye based on the Stiles-Crawford effect. Some of the cones do not obey that. They get light that has been refracted by the foveal pit here. And these cones are a subset of cones in the fovea. They form the focal annulus. These cones are special in that they receive and detect the divergence of and align to light that has been refracted, light that has been split into O rays, shown in blue, and E rays, shown in red. And both of these kind of forms of light, having been gone through the HFL, arrive with shifted directions, and they enter the pupil, not from the center of the pupil, but eccentrically, and in fact, ipsilaterally. And the E rays and the O rays are polarized orthogonally to each other. This is quite different from what one has so far thought. But by doing this, these cones, having this shift alignment direction, they can use the Scheiner principle, and they will know the best focus level for them at their location in the fovea. So here is a diagram of both the fovea of the eye, and shown shaded in gray is this circular annulus. And I've superimposed the graph of the shifted light level direction for the cones in the foveal focal annulus. So the whole macula goes out to here. Within the fovea, there is, within the fovea, between about eccentricities of 0.8 and 1.2 degrees eccentricity, the focal annulus, which does something special. And interior to that, there is the foveola, and from like 0.0 to 0.6 degrees eccentricity, which is specialized, the cones there, for high visual acuity, but they don't know how to focus. So this focal annulus, I propose, is a specialized, not for high visual acuity. It can't do that, but it can do something sort of as an accessory function for the central cones. It can tell the whole visual system how to focus so that the cones in the center of the eye are going to be well focused. And one assumes that when these focal annulus cones are well focused, that the cones in the center of the focus of the fovea will also be well focused. So these findings provide evidence that the focal annulus is not specialized for spatial resolution and visual acuity, but is instead specialized for adjusting the lens accommodation level to focus the eye. So what factors are relevant for this function of this focal annulus? Light dispersion according to wavelength, longitudinal chromatic aberration, which results in the chromatic difference of refraction in the eye, which varies with wavelength. And we have three types of cones: short, medium, and long wavelength sensitive, whose spectrum, spectral sensitivity and photopigments vary. We can use all of that information. Because of longitudinal chromatic aberration, the SM and L cones are in best focus at different wavelengths and at different ocular focal lengths. So in addition, the macula contains high levels of yellow macular pigment, whose absorption spectrum is for short wavelength light, and the macular pigment is primarily going to absorb and affect short wavelength cones. So the lens accommodation mechanism could compare the photoresponses from 2 or 3 cone types and calculate a compromise level between those levels preferred by S, M, and L cones individually. We can show that here. Upper left is chromatic difference of refraction versus wavelength, and you can calculate, transpose that into versus wavelength of focal length, and I've made it that these curves are just the same. We can then combine them on one curve at the bottom, and you have wavelength versus either chromatic difference of refraction or focal length of the eye. So the S cones, their maximal sensitivity is more or less here, and they would like this focus level, which corresponds to this focal length. The M cones have this maximal sensitivity, and they want this ocular focus length or this focal axial length of the eye, and the L cones prefer that. So I am saying that if we have all of these 3 kinds of cones, the focus signals are probably going to be made as a balance compromise between these. Okay, to rest your eyes for a minute, please look at this drawing. It's a painting in the San Diego Art Museum, which demonstrates chromostereopsis. So if you have binocular vision and you look at this painting with both eyes open, you will see that the red colored images appears closer to you than the blue images, which are far away. This is demonstrating longitudinal chromatic aberration, okay? I hope you can all see that. If you now close one of your eyes and look at it, lo and behold, the depth perception disappears. This is a binocular phenomenon. People don't totally understand how it happens, but this is, I believe, related to how our eyes perceive depth and also how the focal annulus functions. We can also describe the role of macular pigment in the focal annulus. There is an entoptic phenomenon called Heidinger's brushes that looks like this. Mr. Heidinger discovered if you look at a blank white background through a plane polarizer and rotate the polarizer, you will see something like this, very small. And when you rotate the polarizer, this will rotate. So this is believed to indicate that the macular pigment molecules in the human macula are tangentially aligned in the fovea, and that they are associated within the Henle fiber layers, which are radial on the fovea, and the macular pigment molecules are somehow aligned perpendicular to either the axons and or the microtubules within the axons. So this phenomenon demonstrates that the macular pigment molecules are oriented with their long axis tangential on the macula. So we can conclude when we know that they're in the focal annulus, there are O rays and E rays. The macular pigment molecules, speaking a little bit loosely, they can only absorb the O rays in the focal annulus. And their absorption of short wavelength light will primarily reduce the S-cone signals of the focal annulus. So I have the suspicion that the orthogonal polarization of the O rays and the E rays gives us some evidence that macular pigment is involved in the function of the focal annulus, and that this is not just an accident. So These findings lead logically to a simple model for the accommodation control in the human eye. It's consistent with extensive research findings. And I have formulated, a detailed model of the steps by which the focal annulus can guide the changes in the level of lens accommodation. I know more than I am presenting here today. The model proposes that the optimal ocular focus is specified as a weighted balance between the signals elicited by light in the subset of S, M, and L cones within the focal annulus. These are special cones. The signals in these different cone types vary according to LCA. These cone signals are modulated by macular pigment absorption of short wavelength light. These cone signals are then processed through the retina by certain cells, normal cells, and they are transferred to certain focus-associated ganglion cells that are there in the eye, in the macula. And the axons of these ganglion cells project in the optic nerve and tract for post-retinal processing of reflex and voluntary accommodation in the brainstem, midbrain, and brain. So the focal annulus in the human eye is likely involved in accommodation to control accommodation in the adult eyes. And if we are, it can also, the same mechanism or analogous, be used when the eye is growing for what's called emmetropization. So the eye of children would hopefully, ideally, grow such that the power of the lens is appropriately balanced by the length of the eye, and everything is changing. But if this is done in a balanced fashion, the eye will not have any refractive errors, but be emmetropic. However, if there is some disturbance in that balance, then the eye of a growing child will become ametropic and like the myopic or hyperopic, or even have high myopia. Because the focal annulus is in the macula and it involves macular pigment, it's quite evident that it's quite likely that disorders of the focal annulus may result in disorders of central vision and the macula, such as macular degeneration, which causes looking at with the macula to give blurred, distorted lines, as which you would expect if the cones are not well aligned. And you could also produce visual problems from viewing some technological devices, such as digital screens and virtual reality displays. So if we want to continue, we can talk about S cones. Here is on the right, the graphs of the total number of cones at different eccentricities. This is at a different scale than on the left. We have just the S cone number of cones. This is 10 times smaller than this. And here are, so this is the normal total cone numbers here, which corresponds to this curve. But okay, the S cone density on this graph would be like this. Okay, so there are not many S cones in the retina or in the fovea. They're at their highest density, at 1 degree retinal eccentricity in the human eye. Again, I don't think that is just a coincidence. I think that is good evidence that the S cones are really important for the function of the focal annulus. So the focal annulus at 1 degree eccentricity suggests, I mean, because that's where the S cones are most densely present, that the S cones participate in focal annulus function. I was interested in the numbers of S cones. When you calculate how many S cones there are in this focal annulus, if you're saying that the focal annulus is that small middle peak of it from this, some 0.8 to 1.2 degrees eccentricity, there are 340 S cones in the total focal annulus. That is, I found amazing. And we can also ask, what is the distance between the S cones in the focal annulus? And we can then calculate based on their density that more or less, if they were equally distributed, which is not in fact the case, but if they were equally distributed, the distance between neighboring S cones is something like 7.3 minutes of arc. That is not very much, but it is, yeah, has some meaning. So work has been done on the rarity of S cones, and it is known that because they are so rare, they can cause what is called aliasing. So aliasing is an effect that causes different signals to become indistinguishable, or aliases of one another when sampled. So because of their rarity, S cones are subject to aliasing at much lower spatial frequencies than M and L cones. Here is a figure taken from a Roorde published paper. And he says to be able to resolve this E, the image must be sampled at enough points. So here is his discussion of the individual cones on the retina, and he's projecting an E onto it. And you want to ask, are the cones going to be able to identify that E or not? Depends on how far apart the cones are. Okay, David Williams, once again in 1981, published an interesting study with including Don McLeod as one of the authors, done at the psychology department at UCSD, entitled Punctate Sensitivity of the Blue Sensitive Mechanism in Humans. He found that some humans, okay, these circles are the presumed S cones based on what he finds, that when these flashes of light are projected as the little dots at different locations in the fovea, sometimes the people would have peaks of S cone sensitivity, some people, that were separated by 10 degrees of arc. And he interpreted this as meaning that these peaks of S cone sensitivity could be individual S cones or clumps of cones. So I have here taken Roorde's drawing and made and repeated it onto making a whole array of cones in what could be the focal annulus. And I have projected onto that S cones, which I show like this, red and yellow so that we can see them. And they are spaced equally apart at 7 minutes of arc from each other. And when you look at this, you realize those S cones are going to have a real problem picking up that E, okay? They are subject to aliasing. And I am suggesting here that that is going to be important for the phenomenon of microsaccades. So humans and some other animals make small involuntary eye movements. People have been studying these and they don't understand even now if these have any function, or if so, what the function is. They are made during fixation. We do not perceive them. We don't know we're doing them. It's involuntary. While we are making them, our conscious visual perception is suppressed, yeah? And so here's a drawing of cone mosaic, and here is an example of how our eyes move over that mosaic for some unknown reasons. Possible functions of microsaccades: to maintain fixation, to prevent visual fading, to explore the fine spatial detail. How big are these microsaccades? So here's one group's description of the probability of saccades, which they call fixational saccades made during fixation. And they have different amplitudes, including 7 minutes, arc minutes of amplitude. And here is another person's summary of how big the amplitude of the saccades is and what frequency. And you will see that the amplitudes of microsaccades made in humans range from 3 arc minutes to 7 to 24 to 40 arc minutes. And I don't know, are some of you realizing what I think is happening? So I am hypothesizing that the microsaccades are made to reposition the projection of important salient visual details onto those rare sparse S-cones within the focal annulus, so that the focal annulus can use the information from all 3 cone types to get a good balance compromise level for the best focal length and optical lens accommodation level for the eye. So here we have, I've projected in yellow here the size of the focal annulus on some diagram of, I guess this is a photo taken with OCT of the individual cones. You'll see in the human fovea, the center of the fovea has no blue cones, but then they are in fact become more prevalent, common at the eccentricity of the focal annulus. So let's imagine we are here taking a little view right here and looking at what's going on at that location within the focal annulus. We project a visual image. We're projecting a blue line like this, okay? And that line of the image gets projected onto the cones, optical apertures of the cones there in the focal annulus. There are not very many blue cones in it. There are many more red and green cones. And I have shown here that just by chance, although this is a blue line, which would really stimulate the blue sensitive cones, none of the blue cones, neither of the 2 that are present there, are going to get that blue light or the edge of it onto their optical apertures. So that object is not projected onto any S cones. What can we do about that? Well, we can make a microsaccadic eye movement. So I have now, the bar of light is in the same position, but we have moved our eyes so that this one important blue cone is now, so these cones have just been shifted here, and the blue cone is now going to be able to perceive that blue part of the image. So the object is projected onto, hooray, 1 S cone. So I conclude that the effective focal annulus function requires that salient details of the retinal image be projected not only onto M and L cones, which are a dime a dozen, but projected precisely onto the rare S cones, which is achieved by making microsaccades. So if we now add the focal annulus, shown in yellow here, because it involves macular pigment, onto the macula, it's going to be located here within the fovea. It's going to, on the visual acuity, be located as lines. They're not very easy to see, but that are not, I mean, they're just beside the highest visual acuity and just peripheral to the area of highest cone density, or as shown here in the lower left, Christine Curcio's beautiful diagrams of the cone density. So the focal annulus is well out of the way of the fovea where we have the highest cone density. So it is, however, going to be providing a good estimate, or it can, of how the eye should best focus the wavelengths. So we have described the foveal oculomonitor cone alignment locus, or focal annulus. It is generated by the coincidence of multiple interdependent components near 1 degree retinal eccentricity. There's the foveal pit, whose walls have maximal slope at 1 degree. There's a thick birefringent Henle fiber layer formed by many long, thin, parallel cone inner axons that are radial on the retina, oblique to incident light. There's a high density of S cones with their optical apertures at 1 degree eccentricity. There is dichroic macular pigment molecules that are within the HFL axons, orthogonal to the axons and tangential on the retina. Incidentally, evidence from monkeys shows that the highly sophisticated focal annulus described in this presentation for the human eye is not going to be quite as sophisticated or as effective in monkey eyes, suggesting that our focal annulus may be a feature that is unique to human eyes. I find that very interesting. So according to the proposed model for focal annulus function, the human fovea contains a remarkably complex and sophisticated neural mechanism that utilizes LCA, compares the signals from 3 types of cones plus 1 macular pigment filter, and calculates a compromised focus value between the different cone signals to effectively control lens accommodation and focus the human eye or eyes. So these findings may be relevant for refractive disorders, which I might mention. There is a global epidemic, not only of COVID-19, but of myopia and debilitating high myopia in the world these days, projected to increase. High myopia is very serious. People have myopia. It becomes so strong that it can lead to a retinal detachment in the one eye. And if they have it in the other eye, they can also get it in the other. They get both retinas detached and they are then unfortunately blind, okay? These findings are also relevant for retinal diseases and conditions particularly affecting central vision or the macula. We know macular pigment is important in macular degeneration, so I suggest it's probably important for AMD, where the Amsler grid lines get being distorted, and the cones only in the center of the macula for unknown reasons wind up dying. So I expect that the focal annulus gets being defective, perhaps because the macular pigment is not doing what it should, and the focusing isn't doing what it should, and everything gets disturbed and the cones die. And it may also be important for novel optical or visual technologies. I wish to express my appreciation and thanks for assistance during the course of this work. Several people from the Vogt Institute, Brain Research Institute in Düsseldorf, also Anita Hendrickson and Christine Curcio for providing the information about the human eye. Shaya Feynman in UCSD gave me some beginning information about birefringence. Martha Rosetta Aguilar in Mexico gave me much more information about birefringence. Barry Sensei, who did the retardation measurements of the human fovea, has also helped me. He is now in Australia. So I thank you for your attention. If you want a Christmas present and you think you would like one of these things, I say to you, I think you already have 2 of them. Okay, thank you.
Thank you very much for this fascinating model. We have time for maybe a couple of questions. So please just unmute your mic and ask directly your question to Dr. Eckmiller. No questions?
I found that interesting, and I have, I'd like to mention one thing that in some ways supports the story, I think, which is that when you try to focus on a small point, it can be difficult to maintain precise focus. Whereas if you have a larger target, it's much easier. And this is something we do something that people know who do experiments with fixation points or targets to constrain accommodation. Accommodation is, in practice, more easily constrained by having a large target available, perhaps especially a textured one, as opposed to a small point. So that's, I suppose, what your focal annulus would predict. So it's one observation that supports the story.
Thank you, Dr. McLeod. Yeah, you were involved in some of this work with David Williams while he was at UCSD.
Yeah.
Yeah.
And also involved in some work that supports your model of photoreceptor alignment, where the cones orient toward the direction where the light is coming from. And we studied a couple of people who have peculiar arrangements of the pupil, one with a displaced pupil and one with a cataract, which had the same effect. And in these cases, the photoreceptors seem to align with where the light is coming from. And after the anomaly in the pupil was corrected, the photoreceptors actually realigned with the center of the pupil where they ought to be. But that's something that's only now, I think, being followed up on by direct physical observation of the photoreceptors.
Yeah, that's very interesting. Thank you for that comment. I might mention David Williams didn't follow up on his observations about the Stiles-Crawford effect in the fovea, but instead he went on to develop adaptive optics imaging for use on the eye. So the very valuable technique of adaptive optics imaging with which ophthalmologists can now look into a person's eye and visualize the individual photoreceptors has been developed by, in large part, his work. Yeah, very useful.
Quick question.
Sure.
I believe Fergus Campbell showed years ago that if you're presented with a blurred picture, you don't have to hunt back and forth, which will tell you which way to focus. You can immediately focus in the correct direction to sharpen it. So would your annulus provide a mechanism for that?
Yes, indeed. That has been one of the puzzles that people trying to understand accommodation have had, because the blur in one direction looks the same as the blur in the other direction. And by my hypothesis, let me see if I can get this back to a much earlier slide here. The cones will individually, okay, that was it there. So the S, M, and L cones each will be using LCA, longitudinal chromatic aberration. They are pointing straight ahead, in a sense, based on the Scheiner principle. Each of them will know what they will know whether they're an S, M, or an L cone based on, where they like, what colors, wavelengths of light they prefer to get a high signal. And they will know, if it's an S cone, it will know I have tried to get this chromatic level of aberration, and I want this focal length, and they will, and the L cones, if will want a different one, focal length and focus level, and the mechanism of the focal annulus, I have so far described the signals getting to the ganglion cells, but all of the focal annulus ganglion cells at all eccentricities have to, I mean, their information has to be brought together, and I believe a compromise will be made between the signals from the S cones, which I'm convinced are important, and the M and L cones to get a compromise level that will be the best focus, compromised focus for the entire eye. And that brings up some very interesting questions because I think that these focus associated ganglion cells are going to be projecting not along the classical visual pathways, some of them.
So you do predict that monochromatic light would mess up your focusing?
Okay. Monochromatic light is going to affect the S, M, and L cones nevertheless, although it's only monochromatic. I do believe that we can accommodate more quickly and more efficiently if the light is a broad spectrum light. I was thinking you're talking about only one eye. If you close one eye, you're only getting the information from one focal annulus. Shall I mention the interesting thing there is that the ganglion cells that are representing the focal annulus in a given eye, they are going to be able to control the accommodation of the one lens in that one eye. Now, based on how ganglion cells are believed, I mean, normally project to the brain, the temporal ganglion cells will stay on the ipsilateral side of the brain and project in the optic nerve to the same optic tract and go to that half of the brain. But the nasal half of the ganglion cells in the retina will, their axons will cross in the optic, at the optic chiasm and project to the opposite side of the brain in the optic tract. So the one lens in the eye the information that's going to control its accommodation would be all of a sudden in both hemispheres of the brain. And focusing, lens accommodation can be a reflex. It happens as quickly as the pupil changing constriction or dilation. And I don't see how you're going to get such a reflex accommodation of the lens if its mechanism has to, its signaling has to go through the cortex. I mean, the people commonly these days believe that the lens accommodation signal goes through the LGN to the visual cortex to extrastriate cortex and motion sensitive areas in the visual cortex. And I actually believe that these focus associated cone axons, some ganglion cell axons, they are projecting differently, okay? I know about it, we don't have time to talk about it. People are, and would welcome discussions with people after the talk, and can be reached at my email. I live in San Diego, and although I am still associated formally with the Institute in Germany. I hope to get some local contact with visual scientists in San Diego area. Thank you.
We have time for one more questions. So if no one has a question, I would like to ask, so you were mentioning that monkeys seems to have less developed focal annulus, and I was wondering if you have any ideas or speculations about like the evolutionary, from like an evolutionary perspective.
Okay.
So
here is one more slide that I had prepared, but I threw it out for reasons of time. This is comparing the distribution of S cones in the human and the monkey macula. On the left, this is S cones immunolabeled, work by Christine Curcio. On the right, we have macaque monkey S cone reflectance shown here. This is work by Hanazono from Japan. So Curcio has found that in the center of the human fovea, there is an area, small area, central about 0.35 degrees. We have no S cones. This triangle shows the area, the point of highest cone density. So there is an, you can define an area on the center of the fovea that has no S cones. And our S cones are present at a larger eccentricity than shown here, but at 1 degree retinal eccentricity. Okay, interesting question. What about other primates? And work has been done on macaque retinas, and they were reflecting, they developed a method to study S-cone reflectance from the monkey macula. So here is the optic nerve, and here is color-coded the reflectance of S-cones from S-cones in the monkey macula, color-coded. And here is from that monkey, quantification of the reflectance from the S-cones, which is indicating how many S-cones there are and where they are as a function of eccentricity, inferior, superior, temporal, nasal. And here is the center of the fovea. And I have put these little lines on it. So there's what they called minimal number of S-cones in the center of the fovea, but it's still quite a high number of S-cones there at the middle. And then there are more S-cones in an annulus, which they say in monkeys is 1.3 degrees retinal eccentricity. So you get that both inferior, superior, temporal, nasal, so it winds up being an annulus here. And the point being that the monkey fovea has S-cones in the very center of the foveola. So the human eye can be understood as being more highly specialized in the region of making a very specialized focal annulus. And I am not the only person who has noted that. So the distribution of S-cones is different in the foveas of macaques and humans. So one would expect that that might affect the function of their focal annuli if such a focal annulus is present in the monkey. I do believe it probably is. And Curcio, in some of her work, reported, and here I quote, the small region at the center of the foveola that lacks S-cones is larger in the retinas of humans than macaque monkeys. Curcio noted that such a difference suggests that the human fovea has been further specialized beyond the monkey fovea for improved spatial performance by excluding more of the photoreceptors, rods and blue cones that do not contribute high resolution. Okay, so, and I would totally agree with her, and I would say that is one of the factors that can make the focal annulus be more perfected. In other words, so my findings support Curcio's conclusion that the human fovea has been further specialized beyond the monkey fovea. And in addition, my findings suggest that this additional foveal specialization in humans may result in improved ocular focusing by the focal annulus. I mean, when you look at the development of the eye, embryologically, first there's just a retina that's undifferentiated, and then out at the center develops this fovea. So it's a very specialized thing with the foveal depression. And previously many people have thought it does nothing. Yeah, it's all, yeah, smoke and mirrors or something. Thank you.
Thank you very much. So we will stop here today, and will be the last talk of this year. We'll start again in January. So thank you very much, everyone. Thank you, Dr. Eckmiller.
Thank you. All right. Bye-bye.
Bye-bye, everyone.
Thank you.